The overall goal of this procedure is to visualize the migration of adult-derived neural precursor cells in the presence of a direct current electric field. This is accomplished by first isolating and culturing subependymal neural precursor cells from the adult mouse. The next step is to prepare Galvanotaxis chambers and transfer the neural precursor cells onto the chambers. The final step is to apply a direct current electric field across the Galvanotaxis chamber and perform time-lapse imaging to observe the neural precursor's migration. Ultimately, the time-lapse imaging microscopy captures the galvanotactic properties of neural precursors in their differentiated and undifferentiated states.
Though this method can provide insight into neural precursor cell galvanotaxis, it can also be applied to other systems, such as a mouse stroke model for the recruitment of neural precursors to lesion sites to enhance neural repair.
Demonstrating the procedure will be Babona-Pilipos, a graduate student from my laboratory.
When beginning this procedure, NPCs must have been in culture for 7 days so that free-floating primary neurospheres are ready to harvest when required. Begin preparing 3 Galvanotaxis chambers by placing 3 square glass number 1 cover slips in a bottle of 6 normal hydrochloric acid overnight. Early on the next day, set up the Matrigel to thaw slowly on ice. After 4 hours, it will be ready to use.